Ever since the discovery of penicillin in 1928, antibiotics containing beta-lactam rings have been used extensively to treat bacterial infections. Beta-lactam antibiotics work by binding to the active sites of penicillin-binding proteins, which bacteria produce, to build the peptidoglycan layer of the cell wall, which is composed of NAM and NAG monomers, connected by short peptide bridges. The penicillin-binding proteins, or PBPs, usually bind pentaglycine bridges of adjacent peptidoglycan monomers to form a unified structure. But when beta-lactam antibiotics bind to the PBP's active sites, the peptidoglycan monomers cannot be bound to the PBP, leaving the peptidoglycan layer incomplete. Gram-positive bacteria have thick cell walls in order to hold the cell together, and without the peptidoglycan layer, the cells fail to maintain homeostasis and burst after a large intake of water through osmosis. Bacteria have grown resistant to the beta-lactam antibiotics in two ways. Most commonly, they produce a special enzyme called beta-lactamase, which cleaves the beta-lactam ring in those antibiotics via hydrolysis, preventing it from bonding with the penicillin-binding proteins, allowing the peptidoglycan layer to be made normally. The second type of resistance is caused by a genetic mutation that modifies the structure of the penicillin-binding proteins. This mutation alters the binding site to bond only to the peptidoglycan monomers, rendering beta-lactam antibiotics ineffective. Scientists are working to combat beta-lactamase by using acids such as clavulanic acid, which tricks beta-lactamase into bonding to it because of its similar structure to beta-lactam. The beta-lactamase is then prevented from bonding to the antibiotic, which is then free to bond to the PBPs. Clavulanic acid is called an inhibitor because of this property. When resistant bacteria die, they release their plasmids of DNA, which is taken up into other bacteria via homologous transformation and endocytosis uptake, thus spreading resistance. Scientists are not the only ones who must involve themselves in combating antibiotic resistance, as a significant cause of its spread is people's misuse of antibiotics. People must follow the doctor's guidelines regarding how large their dosage is and how long to take it to ensure the bacteria is completely eradicated. Also, antibiotics should only be taken when necessary, since they put selective pressure on bacteria to develop resistance and can lead to the development of superbugs that turn into a minor illness into a trip to the hospital.